So welcome for attending this update on that Bala will give on developing methods to isolate islets from improved methods to isolate islets from young donors. So we're really excited that he's agreed to do this. I think from our perspective as a supplier of these enzymes, the two biggest challenge I know all of you face is trying to get more islets out of chronic pancreatitis patients as part of a TPIAT procedure, and of course the young donors. I think in preparing for this and talking to Bala for this presentation, I started to go through some data from UNOS and came across a very interesting slide, if we can get it here. Statistics. I just did it yesterday, and the nice thing from the UNOS statistics is that it allows you to break them out by age group. So you can go up to 34 years old, and you can see that we're seeing in the US a drop in the percentage of donors under age 35. But the amazing thing is we've always had a lot of young donors in the US. So, whether or not, then to be curious in the discussion, people have always seen this, what Bala's seeing or not. I'd be curious because it's definitely increased as a percentage, but it's always been a high percentage of donor pancreata were from young donors. So without further ado, I want to introduce Bala. I've known Bala since 2006. Where he has 32 years experience in isolating mammalian islets, and of course he has a lot of experience in isolating non-human primates, porcine islets, and human islets. In the US, he isolated and continues to isolate human islets for clinical transplant. He first began at Pitt in the US, University of Pittsburgh, then Minnesota, Louisville, nationwide, and now he's at Norton Healthcare in Louisville, Kentucky. He's done greater than 500 islet isolations from chronic pancreatitis patients as part of the TPIAT procedure, and he's published over 250 papers and review articles. So one other question, just one other point. The ground rules are please keep your mics muted. Q&A section will be at the end. You can always make comments or questions using the chat box during the presentation, and this session will be recorded for other people to see, or if you want to look at other aspects of it. So without further ado, thank you, Bala, for agreeing to do this.
Thanks, Bob. Yeah, so good morning, everyone. Thanks, Bob, for the kind introduction and for the opportunity to revisit the nuances of young donor islet isolation. For those new to the islet field, it is important to highlight the unique challenges we face here. Typically, young donor islets are mantled. As you see here, this is a typical mantled islets, which often leads to significant loss during the purification process and a lower overall islet yield, and often leads to failed isolation for clinical transplantation. Again, these are the typical mantled islets. Though the surrounding layer of acinar cells may protect islet integrity, it gives some kind of mechanical protection, but purification will become a challenge. After purification, lots of islets will be in the co-bags mixed with a large amount of exocrine cells. So we lose a lot of islet cells during the purification process. Today, I will briefly discuss the role of proteases in digesting the peri-islet basement membrane and then subsequent release of islets, especially from young donors. This embedded islets are not only problem for young donor, even for adult donors, we face the embedded islet problem. My insights are drawn from both published literature contributed by our esteemed friends and colleagues, and my own experience in processing numerous human pancreases. We have gained valuable insights by comparing islet isolation across age groups in porcine as well as in non-human primate islet isolation. And most notably, some of our most interesting recent advances have come from split lobe experiments. What makes this trisected pancreas model so special is that it allows us to test different variables on the same pancreas. And you always have a control lobe and comparing with the two experimental groups. In one series of experiments, we tested an extreme collagenase dose against an extreme protease dose and using a third lobe as our standard control. We have also used this to test from, for example, 3 different vendors. I know you are all using different formulation from different vendors. So Roche supplies collagenase in combination with the thermolysin. Serva provides neutral protease as a protease. And in Vidasite, you have a BP protease option. So we have also used this to test enzyme from 3 different vendors, and by splitting pancreas into head, body, and tail, and rotating the lobes for every isolation, so that you will have a comparable result. And we were able to get much cleaner picture of pancreas digestion profile using this trisected pancreas model. And beyond just testing different brands, we can also use this split lobe approach to fine tune our islet isolation protocol. For example, we have explored various doses and ratios of collagenase and proteases and compared, for example, parenchymal distension against ductal distension. As you know, parenchymal distensions are commonly applied for our auto transplantation because of the fibrotic nature of the pancreas. We also look closely at digestion kinetics, specifically time, temperature, and the impact of prolonged versus short enzyme exposure on islet health. So those things can be comparable with this trisected pancreas model. In today's presentation, I would like to cover the following key points. The first one is islet loss during purification of mantled islets. That's the number one problem for young donor islet isolation. And then the other aspect is islet isolation with collagenase alone without using any protease to understand the role of protease. So I'm sure it's not a common practice to use just a collagenase alone without protease. So if we use collagenase alone, what is the effect? I would like to really discuss that. Then the role of excess amount of protease. If you use excess or in combination with the other proteases like AM protease, clostripain, or elastase, or high tryptic like activity enzyme, what is the common effect? And how we developed this young donor islet isolation protocol. This protocol we commonly apply for brain dead donor as well as for pediatric auto transplantation donors. And then fundamentally, scientifically, what is the possible reason for obtaining mantled islets in young donor? And another aspect is how many numbers are available, young donors are available, and what is the quality of islets? As Bob mentioned, there are many young donors are available. Many times we don't accept those pancreas because of smaller size in pancreas or because of getting embedded nature of islet cells. So generally, the literature consistently shows that even under standard condition with the different gradient approaches, we recover only 65 to 70% of islets following purification step. That's kind of our standard. This is from a published papers from 830 islet isolations. The average recovery rate was 65 to 70%. However, when dealing with mantled islets, a common issue in young donors, that recovery rate drops to less than 50%, representing a significant loss of viable islets. And Generally speaking, many centers avoid using young donor pancreas. As I said, the small pancreas size is one hurdle. We prefer to get large size pancreas more than 70 grams so that we will get enough islet cells for transplant. Then other aspect is the real challenge lies in the mantle nature of the islets. In young donors, we frequently observe a significantly higher pellet volume, often exceeding more than a 0.4 cc per gram of pancreatic tissue, which is markedly higher than what we see in older donors. So when you digest, let's say you take 1 gram of pancreas and digest and get the cell pellet. So in that 0.4 to 0.5 cc's your cell pellet remaining our extracellular matrix. But only in young donors we get that larger tissue pellet, but in older donors sometimes we get really low amount. The difficulty in separating islet cells from the surrounding acinar tissue may stem from two primary factors: an exceptionally strong physical association between the two, basically islets and the exocrine cells. Maybe this connection between exocrine and islet cell is really strong, or even before enzyme reaches this peri-islet basement membrane area, maybe they are all falling so quickly because of the collapsible ducts in pediatric donors or basically young donors. So because there is only two possibilities. Maybe this connection is strong, or maybe they are falling before even enzyme reaches this location. As you know, this is a closer view of how you inject the enzyme, how enzymes are acting on the extracellular matrix. For example, these are all extracellular matrix. These are all islet cells. And generally, the islets are outside the exocrine lobules. This is exocrine lobule. For example, when you inject the enzyme collagenase-protease through the pancreatic duct, the enzyme reaches the exocrine lobule. Because of the mechanical pressure, basically you break all these membrane and allow the enzyme to come out of it. And so these enzymes basically digest. These are all predominantly collagen fibers, and this is the peri-islet basement membrane. And histologically, you can see this blue color is the peri-islet basement membrane. So again, during the distension phase, the enzyme is infused into the pancreatic duct, eventually reaching the acinar cell lobules. Then resulting mechanical pressure disturbs the acinar cell membranes, allowing the enzyme to reach this area. Many studies reported that islets derived from young donors exhibit superior islet function. For example, when you are having a difficulty of mantled islets, purification challenges, but still, why do we need to isolate islets from young donors? Because they are superior. They have a superior physiological performance, especially diabetes reversal capacity and the insulin releasing capacity are always higher for the young donor islet cells. And when we look at the UNIS data from the last 26 years, a very clear trend emerges. More than 30% of our donors are under the age of 35. This means that nearly 1/3 of our potential islet supply comes from high-quality young donor group. And the young donors are highly resistant to oxidative stresses during islet isolation. And again, it's very important for us to optimize the islet isolation technique to utilize these young donors. Historically, numerous studies have attempted to optimize islet isolation from young donors. For example, study from Dr. Shinichi Matsumoto. In his study, he used injecting concentrated enzyme and tried to get free islet cells. And notably, this particular study from a Milan group, this particular study achieved a significant milestone by successfully isolating a higher proportion of free islets using large amount of enzyme dose. So this is their comparison group. Their protocol utilized dual phase enzyme delivery and initial enzyme injection at the procurement site, like a 240 ml collagenase solution, in the procurement site. Then after transporting the pancreas, additional 300 ml of collagenase solution. But for their control group, it's just after pancreas comes to the lab, they injected 300 ml of collagenase. So that's kind of their comparison. So during the study, the strategy was based on the hypothesis that the ductal system in young donor is kind of they are collapsible, and if the enzyme is injected at the time of procurement, it may preserve the integrity of the ductal system. And this, in the study, they had successfully achieved higher proportion of free islet cells, as you see here, like their purity levels 88% compared to their control group. And during my tenure with Dr. Rita Bottino at the University of Pittsburgh, we processed a significant number of young donor pancreases, including one from a 6-month-old donor. We developed a young donor islet isolation protocol. One of our most interesting observations was the presence of beautifully matured islets, even from a 2-year-old donor. This is comparing 50-year-old donor. So usually the pancreas is small. We got a free islets even from this 2-year-old donor. We developed this young donor protocol. So in our approach, so this is during Liberase era time. So this is not in the recent period where we are using all the recombinant or purified form of enzyme. This is during the Liberase period. So our approach was we prolonged the enzyme distension step for over 25 minutes to allow the pancreas to digest in a static digestion condition. This method initiated the digestion in pan before we bring the pancreas to chamber. One of the oldest islet isolation procedures, before Riccardi method, it's a static digestion method. So you inject the enzyme and you allow the enzyme to cook. So it's a static digestion procedure. So almost we followed that approach before we brought it to Riccardi chamber. So in the Riccardi chamber, other options we did, we put 2 screens. We thought maybe this screen will kind of trim the excess acinar cells which are attached to the mantle islets. And this method did not allow the release of large tissue mass. As you know, during the human isolation from young donor, we see large cell clusters. So having these 2 sieves, it really reduces that large mass. And another aspect is, even if large mass comes from a chamber, extract screen in the chamber and the collection conical, again, further trimmed the acinar cells. This was our approach. The major aspect is this method should allow during this digestion process, we have to very gently rock the chamber. We don't want to really allow the mechanical digestion happening in the Riccardi chamber. We don't want to release large clusters. I think because of the collapsible nature of the duct, the moment you start shaking the chamber, it causes release of large chunky acinar cells from these ones. And we noticed, excellent insulin releasing capacity from the islets, which we isolated using our approach, and then diabetes reversal capacity was high from these ones. So in recent time, Greg Zart from UCSF, our colleague, showed mantle-free islets from young donors utilizing truncated collagenase in combination with the neutral protease from Servo enzyme. Personally, I was thrilled with this result when I saw the result for the first time. The islets were beautiful. There was no mantle islets at all. As you see here, these are all like a kind of 17, 22, 19 years older, except this one. I thought that we cannot get mantle islets from standard islet isolation technique. So I was under the impression that is the nature of the pancreas. We have to alter the islet isolation technique to get mantle-free islets. But Greg Rizard showed a lot of free islets. However, the study proved the fact that tissue dissociation enzyme can change the dynamics. In his procedure, he didn't have alteration in islet isolation procedure except the enzyme ratios. He used low-dose collagenase of 1600 Wunsch unit with a high amount of neutral protease that was 200 DMC unit. So during clinical islet cell consortium trial, this was one of the protocol using 1600 collagenase Wunsch unit of collagenase regardless of the pancreas weight. So you see like a 1600 Wunsch unit and a 200 DMC unit of neutral protease. So many young donor islets had high BMI. That's also is another important factor I want to emphasize. If pancreas, though it's a young donor pancreas, but the BMI is high, for some reason we get mantle-free islets. So maybe the enzymes, they can easily act on the peri-islet basement membrane from pancreas from a high BMI case. Really, we don't know. But generally, but if it is a lean donor, obviously we see a lot of embedded islets. So utilizing trisected pancreas model, we tested extreme doses of collagenase and proteases. For example, we used only collagenase in one lobe and a standard dose of collagenase plus protease in another lobe and increased protease in another lobe. I think this is a very important study. So when you use collagenase alone, what is the effect? When you use a standard amount of collagenase and a standard amount of protease, how's the islet morphology? Or if you use standard amount of collagenase but higher protease dose. So during this time, we see very clearly only embedded islets when we used collagenase alone without protease. So you can make embedded islets using collagenase alone. It indicates the importance of protease. I think really I want to emphasize this particular study. Whenever we use just a plain collagenase, all your islets will be embedded. So what does it mean? So that means collagenase digests the collagen fibers, but in order to digest the peri-islet basement membrane, as you know, peri-islet basement membranes consist of type 4 collagen, fibronectin, and laminin. So in order to release the islet cells from the surrounding peri-islet basement membrane, you have to have a protease. For example, when we use a standard collagenase plus protease combination, we saw some free islets and some embedded islets. As you can see, some embedded islets. But increased protease low, in protease low, we saw mostly free islets. Again, it proves the role of protease in digesting peri-islet basement membrane. So based on this observation, so we further continued our study. After validating the trisected pancreas model in older donor, we wanted to test different enzymes in young donor. In this particular study, we first characterized young donor collagen and basement membrane composition. We measured high collagen content in the young donors compared to older donors. And we noticed that type 4 collagen and laminin levels were higher compared to in younger donor compared to older donors. So this is a kind of proof that the extracellular matrix composition is different from younger donor and older donor. We So we prepared 3 enzymes based on this observation. We prepared 3 different doses of 3 different enzyme dose. Basically, extreme dose of collagenase. For example, this is your standard dose. 21 unit is sufficient to digest the pancreas. In order to test extreme dose, we used a 61 unit. And for proteases, just 2 DMC unit is more than sufficient if you are using a neutral protease from server. And so we used a high dose of 10 DMC unit. Similarly, higher thermolysin unit also we used in order to test the extreme doses. So the way we did in a split lobe model, 1 lobe standard dose, another lobe high collagenase and standard protease, in another lobe higher protease but standard collagenase. When summarizing our result, we observed free islets in increased protease dose. So this is the combination, standard enzyme combination, increased collagenase combination, and increased protease combination. We saw many free islets whenever we used the increased protease combination. And ultimately, we have to test, since we used a higher protease, their islet morphology, even islet morphology at the scanning electron microscopic level, and then insulin secretory capacity in dynamic glucose challenge, and diabetes reversal capacity in diabetes-induced mouse model. So we saw that using higher neutral protease, we did not have major problem in getting our islet quality was not compromised. That was kind of our observation from this particular study. Then, so maybe we thought we are getting free islets because it was a split lobe. So during split lobe, we are using a small lobe. Maybe enzyme distension step is so easy whenever we inject the enzyme in a small lobe. So, but we kind of validated our procedure in a regular lobe, full lobe pancreas, and tested with the different all the enzyme combinations from Sharva, Roche, and Vita-Site to double check are we getting a similar effect when we use a higher protease, regardless of whether we used a neutral protease or thermolysin or BP protease. So these results were well presented, in this paper, and in based on our experience using our young donor protocol, we applied and successfully utilized our young donor protocol for our islet cell auto transplant. When we do pediatric islet auto transplantation, if we get mild fibrotic pancreas, that is minimal change fibrotic pancreas, that's going to be an extreme challenge for us, because for pediatric islet auto transplantation, we want to provide smaller pellet. But if islets are embedded or mantled, we cannot purify those islet cells. So we end up large pellet volume. That's going to be a real problem for infusing into the portal vein. But at the same time, if your pancreas, even if it is a pediatric donor pancreas, if it is a moderate fibrotic or severely fibrotic, we don't need to do these adjustments. So there, we can use less dose of protease, because even if the islets are embedded in those scenario, we are not going to get large amount of tissue pellet. As you know, in moderate and severely fibrotic pancreases, your parenchymal cell mass is very low. So we are not going to get large mass. So there is no purification is required. So during that period of time, during that scenario, whenever we see moderate and severely fibrotic cases, we use less protease, because we need more collagenase to digest the large amount of matrix, ECM matrix present in those pancreases. So we use a large amount of collagenase, and we, even if islets are embedded, that's not a problem because it's a small pellet. If it is the other way around, if the pancreas is mild, that is minimal change pancreatitis, we have to be extremely careful in selecting the right protease dose. So now, based on all my observations, I would like to suggest, this is kind of my approach, So this is the main aspect I want to discuss with you all. Let's say you have a pancreas from a 21 years old from a female donor, and the height is only 5.5, and body weight is 50 kilogram. So your BMI is low. So in this, when you process this kind of pancreas, you know that you are going to get large amount of embedded islets. So based on your standard islet isolation protocol, that's going to be your outcome. Without even doing isolation, we can see based on the donor information, we can exactly tell that's what it's going to happen. Because if at least if the BMI is high, if the body weight is high, for some reason we are getting some free islets, maybe not 100%. Definitely, it will be better than your 100% embedded islets. So, this is a classical example again. When you have a pancreas like this, what do you need to do? You have accepted the pancreas, your pancreas has arrived to the lab. Now you need to alter something. What you can alter? I know if it is a clinical program, either any clinical program, you have a definition of, you define the enzyme dose. You cannot simply use a large amount of collagenase or large amount of protease. So you need to, however, you need to adjust to the maximum what is allowed in your clinical protocol. So you have to pick the large amount, whatever the highest dose of your protease. That's your first step. And I definitely, the standard enzyme distension duration is 12 to 15 minutes. But according to our protocol, we try to prolong the enzyme distension. So prolonging the enzyme distension is not a harmful process, because basically you are initiating the digestion in pan before you bring the pancreas to the Riccardi chamber. So that shouldn't be a problem. So if your protocol allows to go for 25 minutes up to 30 minutes, you can increase the distension duration. And finally, when you place the pancreas in the Riccardi chamber, try to minimize the mechanical digestion. Just gently rocking the Riccardi chamber, that alone is sufficient, because you have initiated a digestion in the previous step. Here, it will allow to release only the free islets, not the large cell clusters. So this is kind of, re-emphasizing, increase the protease, prolong your enzyme distension step, and slow mechanical digestion procedure. So I know Bob mentioned multiple times, Bala, this technique, whatever you published, it is not very effective when other team members tried. So I think, so when I asked the next question, did they do all these three steps? If you don't do all these three steps, you may not see the real effect. So this is the type of pancreas you have it, and you have to make some adjustments. And these are the adjustments, we are suggesting. In case, for example, you have used a higher protease, but if you start shaking your chamber vigorously, basically you are initiating a mechanical digestion, then there won't be real effect. Same thing, if your procedure doesn't allow you to go for a static type of digestion, again, you may not see the effect. So I think this is kind of my suggestion for you all to try, whether you see any difference. You may see a difference. And again, when it come to islet isolation protocol in different labs, there are a lot of variations. Some labs, they inject warm collagenase instead of injecting cold enzyme. So then there is a real variation in that. Some labs using the Riccardi isolator instead of handshaking. And there, the tube length and the enzyme volume is kind of different. So that is another differences. And many labs, they use a hand syringe enzyme distension or using the peristaltic pump or automated pump system to inject the enzyme. So again, that's additional variation. And some labs, they start the digestion process after soaking the pancreas in Riccardi chamber for at least 5 to 10 minutes. Again, reaching 37-degree, our enzyme collagenase is active at the maximum activity is at 37-degree. But in many labs, we noticed even it takes longer time to reach 37-degree. So that is also another variation. So, but based on our kind of recommendation, these are the main ones. And in general, I would like to kind of really summarize what are the protease combination apart from our standard neutral protease or thermolysin or BP protease we are doing. So we have tested the effect of cladribine, AM protease, and elastase. I think combination of protease is also important for digesting the peri-islet basement membrane. Historically, when Clostridium histolyticum enzymes, it has all kind of impurities along with collagenase, neutral protease, high tryptic-like activity enzyme, cladribine is present. So those enzymes also really necessary for digesting these type of young donors. It may or may not necessary for our older donor. And other aspect is the embedded islet problem. It is not only seeing in younger donor, even in older donor, we see that the embedded type of islets during the later phase of the digestion. So for that, that leads to, again, low islet recovery as well as, it leads to failed islet isolation. So when we did the analysis in clinical islet transplantation consortium data, the failed islet isolation or the success was only 50%. 50% of the time, many centers could not transplant the islet preparation. So when we analyze the data, predominantly we see the 50% of the time, the failure was due to purification issue. So again, to summarize, optimizing digestion step of peri-islet basement membrane can decrease the mantle islets. Again, increased dose of protease, whether you are using neutral protease or thermolysin or BP protease. With the standard dose of collagenase can be utilized effectively to obtain mantle-free islets. And one caution, whenever you are using thermolysin is a very aggressive protease when compared to neutral protease and BP protease. So when you are using thermolysin, we have to be extremely careful in stopping the digestion at the right moment. Whenever we see the islet morphology after isolating with thermolysin or neutral protease or BP protease, we see better islet morphology with the neutral protease and BP protease when compared to thermolysin. So again, it's very important to carefully stop the digestion process. And the enzyme enhancing enzyme exposure and the minimal mechanical digestion in Riccardi chambers are necessary, along with changes with the enzyme. And as I said, thermolysin can damage the islets if it is not monitored. And the combination proteases are definitely effective in providing free islets. And another aspect is modified purification approaches are necessary. So maybe I can give an example here. When we do auto transplant, we know that we are not going to large amount of tissue pellet. At that scenario, you can make all your islets embedded, and those embedded islets, when you do manual sedimentation, those islets, because they are large clusters, they will sediment very quickly. On top of that, there is a large amount of exocrine cells, floating exocrine cells, that you can easily eliminate. So that's a manual purification approach we use for some of our pediatric auto cases. Because when we have if we don't see free islets, generally they don't float. As you know, exocrine cells are higher, their densities are higher. So islets are generally, they tend to come up. But if islets are embedded, so they will go to bottom, still you can eliminate large amount of exocrine cells. And our ultimate approach is, if we have a way to transplant large amount of tissue pellet for our transplant, that will enhance, or basically it reduces islet loss by purification, because even during purification we have to expose the islet cells to gradient material, and it consumes additional 1.5 hours for our islet isolation procedure. If you are doing two purification, again it's additional hours. So but if there is a way to transplant large tissue pellet, by transplanting into the secondary site, then we can transplant large amount of islets. I'm sure every lab now, the enzyme steps are well standardized regardless of the donor age. We see very minimal undigestion, and we see large amount of islet number after digestion step. So the digestion step is well controlled in every lab. So we see large number of islet cells. But if we can transplant almost for example, like a 30 cc cell pellet or 40 cc pellet, so almost every clinical islet preparation will be transplanted. Because today, till today, the success of islet isolation success is only 50% throughout the world. Many islet preparations are not transplanted because of the low islet mass or purification issue. I think I would like to really acknowledge all the members who worked with me in Pittsburgh, Minnesota, Louisville, Norton Healthcare, Nationwide Children's Hospital. Without all our team members' help, we wouldn't have learned all these things. And thanks to all the enzyme companies, they cooperated very well with us, and we provided our feedback. They produced all recent modern enzyme, GMP enzymes, GMP grade enzyme, purified form of enzymes. And I'm thanking our organ procurement organizations, especially my Kentucky Donor Organ Affiliate and funding agencies. I think with this, I stop. I would like to discuss this topic with you all. If you have any question, please let me know.
Okay, great. Thank you, Bala. That was a wonderful presentation, and it's great to see the historical aspects of how people have tried to address this issue. I can just there are a few questions that I'll just go through. Hannah Schultz asked, what do you consider standard dose of collagenase? And after we go through these ones on the chat, you can unmute yourselves and ask questions as well. But maybe answer Hannah's question first.
Yeah. Like a 21 unit is kind of, it's a right amount. We tried 12 unit to digest the pancreas. Only if the enzyme is powerful, like example, recombinant enzyme, you may able to digest the pancreas with the 12 unit. That is the lowest. 20 is the kind of standard collagenase dose.
Yeah, I can make a comment on that as well. I think there was interesting studies, because what Bala just mentioned, he was getting with our recombinant collagenase enzymes using 12 Wunsch units. And when you went down, Bala, to 12 Wunsch units in the natural purified phytocyte enzymes, you cut the yield in half. Doug O'Gorman which it's where it's published actually in abstract form, has showed that he can use 16 Wunsch units of either natural purified collagenase, or that's how we sell our recombinant our collagenase HI product, our recombinant collagenase product, and you're getting equivalent yields, and you're getting a better in vitro function for in glucose stimulated insulin release test, and those you can find in those abstracts you can find in the IPETA meetings in 21 and 23. And Sui asked your question about maybe basically Wang Xing, if you're on, maybe you can say it, because it's a long question. I don't want to misrepresent anything. So if you can unmute yourself if you're still on, it'd be great.
Thank you, Bob. This is Wang Xing Cui. Again, Bala, thank you. This is an excellent presentation. I learned a lot. According to your presentation, in the content, you mentioned multiple times the parameters are variable. Then this decision-making process is quite subjective. You need intensive experience. We work in this field for many, many decades. But this still didn't solve subjectivity, so subjective decision-making. So then what my question for you specifically is, now is the 21st century, AI is evolving, and machine learning is very rapidly developing. So many areas, what I observe, they're using a machine learning AI-assisted QC testing system already developed. So my question for you also, I want to propose to this community, we should work on the AI-assisted human islet isolation system, try to make subjective decision-making procedure to convert it to the objective. Those are the decision-making metrics. What do you describe? The digestion time, the pancreas structure, the BMI, those parameters, we can ask AI to the major heavy lifting work. So what do you think? I just want to get your opinion. Thank you.
Yeah. Thanks, Sai. That's a very good suggestion. But in order to, as you said, like in order to avoid, what is the best time to switch your digestion parameter? So in order to avoid all those things, there are two things we can do. I think, in Riccardi islet isolation method, it's an excellent procedure, but in two places we have issues. One is the enzyme distension time. Some labs 8 minutes, some labs 20 minutes. So how do we really decide? We need to switch. You would stop injecting the enzyme at 12 minutes or 15 minutes. It should be a dynamic. So for example, like let's say you start injecting the enzyme whenever pancreases, doesn't hold the enzyme. So that's kind of your actual time to stop your enzyme distension. So it should be a dynamic process. Same thing for your assessment. Let's say you start digesting the pancreas. Whenever you see islet cells, if you have a system, instead of stopping your digestion, you start collecting the islet cells and allow the enzyme to go back to the circuit. So it's a dynamic system. So whatever the free islets are coming from the chamber, you have a system to collect, but your digestion is ongoing. So that means even blindly, even without taking any sample, you can get a good islets because you are not stopping your digestion. Because of the enzyme recirculation, it allows the digestion of undigested portion of the pancreas. So we are also making some changes. We are in collaboration with the bioengineers, even BioRept team, to make this modification. So yeah, it's a good suggestion to use AI tool.
Can I make a comment on the AI? Because as a person who's always seen these things coming, and I know AI is coming, the issue that you have to do it right, you have to have great data sets. And when I wrote a review article about the evolution of enzyme requirements from the enzyme side, most of the enzymes we used in other reports weren't that well defined, and we don't have a big data set. So if we're going to do that, we're just going to have to figure out a way to get the data set so the AI can tell us something. And it can find relationships that we can't see, but it all depends on the data you feed into it.
Can I add one more comment, Bob?
Sure.
What I want to direct this way, data-driven. I completely agree. We need to collect more data.
Yeah.
One experience I want to share with this group, for example, donor selection. When I got the phone call from the OPO, I can get the donor chart, make AI to make a decision. Let AI make a decision.
So that's a different story. Yeah.
Whether I can select, I should accept or decline those the acceptance criteria, exclude criteria already train the machine. So the machine will help you to make a decision. Then related to this topic as well, if we have all donor information, AI can tell you what enzyme we should select. Then if we have image to tell the AI, they will say what the wrong, what the step you need to adjust. Prediction, what I'm saying.
That's where I'm saying that's a little hard because we don't have enough data. And to be frank with you, being in this field as long as I have, I think the issue that should be emphasized is we should be using several enzymes. And there's hardly any studies done. Bala, I mean, has done some work on that, but you need to use complementary proteases. And I think we see a hint of that in the periodic reports that come up when people find high cluster pain values help. So it's something I think that the suggestion you make for donors, the way people do it, I think that's a superb recommendation. Getting down to the islets, it's going to be, it's just, we don't have the data sets. My opinion.
Yeah, I think like a simple tool, to assess the donor quality, especially
right.
Tatsuyakin reported, body surface area plays a major role in knowing the size of the pancreas. Instead of BMI, body surface area is really a key factor in deciding the right donor. That's in my opinion for clinical transplant purpose.
Hi, Bob. This is Bashu from Dallas. Can I ask you a question?
Surely.
Yeah. Hi, Bala. Thank you so much for a very excellent presentation. I think you have this knowledge and experience that's very helpful for the field. We have been, in this field for more than 20 years, and we continue to experience the same issues that you were discussing. Like, for example, when you have young donors, whether it is for autologous isolation or for even from deceased donor isolation, we end up having so many mantled or embedded islets, which are difficult to purify, and the pellet volume becomes very high, and it's become a challenge to decide how much to infuse into the patient because the patient may experience portal vein thrombosis. So the changes that you have suggested, like for example, double screen, changing the neutral protease concentrations are very helpful. But I have a small question. That is, when we talk about TPIAT procedure at our center, the cold ischemia time is usually less than an hour. I noticed that in most of the studies that you published, your cold ischemia time is around 6 to 8 hours or, a little bit that range. So does it play a role? Like, shorter cold ischemia time, is it changing the enzyme required or the release of islets from the pancreas? Thank you.
Yeah. Thanks, Dr. Nazir. Yes, regardless of the cold ischemia time, in order to avoid mantle islets, so we have to adjust the dose for sure. So when we do brain-dead donor, obviously the cold ischemia is longer. But for younger donor, even with the longer cold ischemic pancreas, we see a lot of embedded islets. Even in that scenario, we have to increase the dose. I know some labs, they are experiencing clumpiness, or maybe the proteases causes more damage to the acinar cells. But no matter what, I think those are all addressable problem. But if all islets are embedded, you cannot really do a transplant. I think that's the fundamental issue.
If I ask one more question, see, when you talk about chronic pancreatitis patients, many of these pancreases also come with fibrosis. They're fibrotic, they're calcified, like that. So your presentation was more focused on the age, young donor, but young donor plus fibrosis, young donor without fibrosis, if there is a patient with genetic etiology, we usually notice that there is not much fibrosis, whereas with other conditions there is. So what kind of changes do you recommend in this case?
Yes. So for younger donor, for our auto transplants, only if the pancreas is mild fibrotic case, you call them like a minimal change pancreatitis. Only in that scenario, you require all these young donor protocol. If pancreas is fibrotic or even moderate fibrotic, we don't require to do all these things, because at that moment, our focus is digesting the pancreas. Even if the islet cells are embedded, it's not a problem, because we are not going to get large tissue volume. So there won't be any purification, so we don't lose islet cells. So our focus is only digesting those pancreas, using more collagenase, even less protease for those cases.
Thank you.
Yeah, Bala, we have a question from Ahad Hamed. And do you think 20 units is enough if the pancreas is severely fibrotic? How do you assess whether the enzyme units should be increased or decreased? How do you determine the right dose? This is an interesting question, as we know. I mean, this has not really been explored. I know you did some work you may comment on by increasing the dose from your new enzyme mixture that you used at Minnesota. But I'll hand it over to you.
Yeah, I think in our batch production record, we have a very clear definition of what dose we have to use for a moderate fibrotic pancreas and mild fibrotic pancreas and severely fibrotic pancreas. So our max, based on our split lobe experiment, when we can go up to 61 unit per gram. So that's the maximum dose, but generally we try to so the range for the severely fibrotic case, so it should be more than 31 unit. That's the minimum dose. If your protocol allows to use 50 units, yes, definitely we can use.
Yeah, and I mean, that's a lot of enzyme, but you don't need as much. Let me ask you the follow-up question based on your Minnesota experience. You probably don't need to use that much protease. And I know people at Oxford had a report that you actually could cut the protease in half and still have no effect on islet recoveries from a chronic pancreatitis patient. And I'm assuming that's because by the time, if it's that severely fibrotic, really there's not a lot of acinar tissue left. So exactly. It's just really getting the finding a way how to use the appropriate collagenase. So I think this is a real rich area for evaluation, to look at different mixtures of different classes of protease for studying this issue. Because would you agree with me? The protease probably doesn't play a role.
Yeah, as I mentioned, if for autologous transplant, for moderate cases, severely fibrotic cases, you don't require large amount of protease at all. Our focus is just digesting the collagen fiber. One additional comment is, the newer elastase, there is a large amount of elastase. Histologically, we see elastase presence in those severely fibrotic pancreas. Maybe that's the one kind of holding the pancreas. So that's the reason maybe we see large amount of undigestion sometimes. So to prevent that, maybe we have to use elastase enzyme along with collagenase instead of other protease.
And I can comment, if anyone's interested in playing around with this, we do have a purified elastase that we can share with you if somebody wants to try this, because it was actually, we wrote a collaborative grant with Bala years ago, and one of the aspects was to try elastase. And my colleague Francis Dwellett ended up just doing it anyway and purifying it. And I think it's an interesting aspect because it's, you know, as Bala said, it may be something that would help release the islets better from these fibrotic tissue. Tatsuya.
Hi, this is Tatsuya. Thanks, Laura, for the great presentation. I have a just totally different idea about getting a free islet, not trapped or not embedded islet. So we may have to focus on selective lysis of acinar cells. So if we can kill or lyse acinar cells selectively, we can solve the problem. So the one thing is we found that the acinar cell express ABH blood type antigens, but not in the islet surface. So for example, we add the anti-A blood type antibody and add the complement, then we theoretically can lyse acinar cells, not the islet. Then we can maybe get a free islet. So probably someone, not me, but someone can proceed such a direction, totally different direction. So
Yeah.
just my idea.
Yeah. Thank you, Tatsuya. You have a large experience in this. So to answer to this question, we tried two approaches. One is, there is a compound called D-L-ethionine. D-L-ethionine is known to damage the acinar cell. There is a lot of studies are done in animal model. If you constantly inject D-L-ethionine, it completely kind of like lyse the acinar cells. You will get only islet cells, in the pancreas. So we tried that approach, but that compound was very ineffective. When you are using ex vivo, maybe it may be effective when you inject into the animal, but it was not effective externally, when we use the pancreas. That's one of the approaches. Then second approach in University of Minnesota, we did a series of isolation. So you take the pancreas, first inject 37-degree warm plain HBSS. HBSS is the one you use to prepare your enzyme solution. So no enzyme, just a HBSS alone. It's a 37-degree warm. So when you constantly inject with your mechanical force, you kind of stimulate or break the acinar cells. At the same time, you are not digesting anything. Basically, you activate your endogenous protease in every acinar cell. And it's a 10 minutes of warm enzyme distension followed by your actual enzyme. So with that approach, we could see elimination of large amount of acinar cells. They are not like a real particle. They are damaged. Our pellet volume was always small with that approach. But for some reason, we didn't really continue that study.
I wanted to ask Bala, at least when I've talked to people in the past about this, you can recover the embedded islets obviously at the bottom of the density gradient. They're heavier than anything else. And what my understanding from people prior conversations is if you kind of wash them and try to redigest them, you can't redigest them. You can't, they're stuck, right?
Yeah.
And
Yeah,
you don't, no one knows why, right? I don't understand why this.
No, maybe no, I don't think so. Really, anyone studied, or experimentally carried out those kind of experiment, Bob, like a redigesting the pancreatic digest with another protease.
Well, with just the embedded islets, just the things that go to the bottom of the gradient, right?
Yeah,
Because that would be enriched in embedded islets, you would think, right?
Yeah. So anytime you get embedded islets, it's an older donor or younger donor, kind of like doing an exposure to some kind of protease, or even like I think people tried even using a trypsin, exposed pancreatic digest to trypsin, and I think some other, I don't remember, like some other proteases, cocktail of proteases are available for molecular studies. People use that. We don't know.
Never worked. Didn't work.
Yeah, the studies are not conducted like a real experimental setup. We try to use it, but we don't know the real effect.
Okay, but it would be an interesting approach, right?
Definitely.
Right. And let's see, we're getting a little over. Let me just one clarification. I'll represent a question Paul Johnson asked you before we close. And this is something I can understand why he asked it. When you presented this data earlier at the NIPETA meeting, he asked the question of how do you define embedded versus mantled islets? And I understood why he said that, because in the original recorded paper, the mantled islets were ones that formed a crescent around the rim of the islet. And I was just wondering if you could comment on that. I mean, is there a distinction anymore between what you call mantled versus embedded, or should we keep that terminology, mantled, as being a rim and versus, or a small rim around it, versus embedded is like what you've shown?
Oh yeah, you are yeah. Dr. Paul Johnson, I think even he himself answered to this question. Yes, there are two different types. Embedded islets are definitely different. You may see a large exocrine cell in the middle. There may be a pancreas, even some small attachment, to the exocrine cell, to the islet cells with exocrine. They are kind of embedded islets. But mantle islets are really different. They are like, as you said, like they are like a well-round. There is a small rim of acinar cells in those ones. But we see both. From young donor, even from older donor, we see both, during the digestion process.
All right. So if there's no further questions, just before we end, this is how we started, and Bala was the first one who gave a presentation in November of 2020. I welcome anyone to contact me if they have an issue that you think would be worthwhile to discuss as part of the islet community. That's what these webinars are being held for, is just more educate ourselves on how we can improve human islet isolation. So with further ado, I'll thank Bala again for a superb presentation, really first class. And because this is, as all of you know who attended this is a really critical issue that you all face. So thank you, and anyone interested in giving a webinar, contact me, and this will be on our website as a recording if you wanted to pass it on to others. Thank you.
Thank you all.